Well, hello, welcome everybody. I'm Vanessa Reyes from the WILDLABS team based at the Wildlife Conservation Society in Argentina, and I'm going to be your host today. So welcome to June Variety Hour. This month's Variety Hours has a special ocean theme since June the 8th was the World Ocean Day. So we thought it would be a great opportunity to highlight some of the exciting work happening across marine conservation technology and in the marine environment. So we have some great speakers lined up today, including speed talks from Aden Ip, Matt Pickett, Alasdair Davies, and then a long talk from Kalindi Fonda. So now I'm gonna hand it over to Aden. No lab, no cold chain, no bioinformatics, real-time eDNA species identification from a backpack.
So hi everyone, happy to kick things off as a first speaker. So I'm Aden Ip. I am a research scientist and a principal investigator at the eDNA Collaborative at the University of Washington. So my talk today it's mainly about environmental DNA and conservation applications in the marine world. First off, environmental DNA. I'm quite certain everyone at least have heard of this term before, but very briefly, every organism sheds DNA into the environment, into the surroundings, into the soil, water, and even the air. And if you can collect it, you can identify what species are present without ever seeing or capturing the animal. And to me, I think this is an incredibly powerful conservation application too. And the barriers to entry have always been high. So theoretically, whenever you want to do eDNA work, you need like a fully, functioning lab, cold chain storage, and a kick-ass bioinformatician. So right now we're trying to navigate around this, high barriers. We're trying to lower the barriers of entry to build a system that reduce the reliance on these things. So this is what a typical eDNA workflow looks like for many people today. First you collect an environmental sample. Most of the time it's a water sample. You freeze it and then you ship it to a lab. Somebody else extracts DNA. Somebody does a PCR and then sequence on a fancy machine. And then a coder or bioinformatician runs it through computational pipelines that's like a black box. You don't really know what's going on. And then weeks to months later, you get a species list back. And by then you have left your field site, immediate follow-up is not possible. You also can't adapt your sampling in real time. And that sometimes the information might arrive too late to be, readily actionable. And for most eDNA teams, I think the bioinformatics alone is a deal breaker because you really need someone who can code, who can assemble a local database that's relevant to the area that you are trying to survey, and also who can troubleshoot a pipeline and talk to the computer. And most field teams don't have the person. So my team right now, we ask this question: what if we remove all of this, restrictions and simplify everything? Can we still get it to work? And we have actually designed a fully portable prototype that fits into a single backpack with no cold chain and also automated bioinformatics expertise. So for the moment you collect a water or air sample, you can see species names appearing on the screen with a turnaround time of about 4 to 6 hours. This is a recent breakthrough from my team. And I think that is the fastest eDNA meta barcoding results turnaround at a community level reported anywhere. So we have also designed improvements like in the wet lab, like you fill the water sample, you extract, amplify the DNA, and then you simplify protocols with less reliance on cold chain and load it on the nanopore sequencer, which is here over on the right, is the size of a stapler. And yeah, everything runs on batteries and solar power. So all you need to do is to look for a sunny place, and then your species names will appear as your environmental sample is being sequenced. And we have been trying to stress test this over several deployments. We deploy it at extremes of the infrastructure spectrum. On the left, we have started with DNA barcoding on a coral reef research cruise, and then we moved on recently this year to do this whole workflow across the Western Pacific from Philippines to Guam on the research cruise. We did real-time eDNA barcoding, and we detected vertebrates, invertebrates, even textiles are associated with deep sea hydrothermal vents or processed at sea. And then on the right, we also ran this workflow in the Amazon rainforest. We picked up Plasmodium, which is the malaria parasite from canopy eDNA swabs. And also with the same workflow, same backpack. In fact, starting today, I'm going to try to deploy this on an offshore island in Washington State in the United States with no power, no lab, and no connectivity. And then we'll do water and air eDNA process entirely off-grid on solar power and batteries. So that being said, we're also trying to remove the last barrier, which is, sometimes we don't even need to sample water at all. And because sometimes there's really no water to sample, or it's just too dangerous to sample the water, or it's just impractical. But at least the DNA is still in the environment, and most of the time it's also in the air. It's recoverable from the air. So water consistently, throws DNA upwards. There's splashes, there's ripples, there's bubble bursting, and that aerosolizes waterborne DNA into the air. Terrestrial species also shed DNA in the air directly. So air eDNA can capture both aquatic and terrestrial biodiversity in a single sample. And we have published a study. We did this test, proof of concept study over a salmon spawning creek with passive air eDNA samplers that require no power, no pumps, and cost less than $10 each. We hang up, come back the next day. And from the same paired air and water sample, we found 35 vertebrate species across air and water. 40% showed up in both media. And you see like even fish in the air and also mammals in the water. It's pretty cool. And also the airborne signals track with actual fish counts week by week. So now I just wanted you to picture if you pair that with a backpack system, and like $10 sampling tray, you can do field sequencing and gives you species names in hours. And that's really amazing to me. And you can actually establish a monitoring network that they can put in places that are too remote or too dangerous or too expensive to reach in any other way. So with that said, we built this system, which is great. It's automated, it's off-grid, but I realized that I can't scale it alone. I'm actually actively looking for field partners. If you're interested to apply eDNA, you manage a protected area, a coastline, or anywhere you need biodiversity data that a lab can't get to, I'm interested in collaborating to bring this backpack to your site and run it with a team as a real deployment, customized, to answer your questions, detect your species, and look at your data. I'm also looking for engineers because this system works in the Pacific Northwest. The biology is working, but I have not looked into thermal management, into tropical heat, weatherproofing for monsoon seasons, and all the shenanigans. Also, beyond species detection, we're also moving on to try to resolve individual animals from seawater. That's a longer conversation, but I'm happy to take this offline if anybody wants to know if you can do eDNA and population genetics. I have to jump off a few after this, but if anyone has more questions and want to collaborate more, just feel free to drop me an email or scan a QR code. You get my email. Thank you.
Thank you so much, Aden. We have a few minutes for questions. I love this presentation because we recently published Talia will mention about that later, but our State of Conservation Technology survey findings suggest that many practitioners are really excited about eDNA, but still find it difficult to implement in practice. So I think you have shown how you remove many barriers, and I wanted to ask you, what do you think is the biggest thing holding back wider adoption today? And if this has changed now that technologies like your portable sequencing is becoming available?
I think there are many, it's a multifaceted problem, but I think the very first thing is sampling. Like people don't know how to sample eDNA. And also the frequency, how many samples you need how many samples do you need to take to detect the target species. So that actually requires collaboration with relatively experienced people with eDNA. And also, I think that's the main problem. And then bioinformatics also spooks people out. So I think that's the two main things that are the main barriers, because the wet lab part can actually be outsourced to commercial companies, but that will be a time lag of like 2 to 4 weeks. So portable sequencing is great because it's going to be 4 to 6 hours, like I said. Yeah.
Thank you, Aden. I think we can. Matt, do you want to ask your question? We can make time for one more question.
Yeah. Aden, two-part questions, Matt Pickett. Is this commercially available or is something that you're creating, in the lab and there's one of one? And I missed what you said about saltwater. We have a upcoming collaboration down in St. Martin to do some eDNA work. And right now the plan is just to send it to the lab. But wanted to get your thoughts on that.
Yeah, it works with any workflow, air, freshwater, saltwater. We just have not done anything with sediment. It's not commercially available yet. There are plans to make it commercially available. We just finished assembling a prototype like 2 weeks ago, and we are doing like our final round of stress testing literally today. So I'll keep you in the loop. We can, keep in touch over email when I have more progress on getting it up down the commercial chain.
Okay, we need to continue. Thanks, Aden. I see Kalindi has a question, and there are more questions on the chat. So please, if you can answer them on the chat, that would be great. So
sure. Thanks, everybody.
Thank you. Our next speaker is Matt Pickett, and he's going to present using Edge AI-enabled UAS to support response and rescue of cold stunned sea turtles. So Matt, if you're ready.
That's a tough act to follow. That's pretty impressive, Aden. I'm very impressed. I'm Matt Pickett with Oceans Unmanned. We're a nonprofit dedicated to using unmanned technology or conservation technology for environmental research and monitoring. We've been around for 10 years doing different unmanned system operations kind of around the world. Today I'll be talking about a project we're working on with Texas State Aquarium and Ocean Wildlife Institute. The problem they came to us was with cold stunned sea turtles down in Texas. When cold fronts come through there, and the sea temperature gets down below 50 degrees Fahrenheit, turtles can become cold stunned, which means they become immobile and they have the possibility of drowning. So right now, the way they respond to that is kind of opportunistic overflights, or they send out fleets of research vessels, recreational vessels, and just kind of hope they stumble across these turtles while they're cold stunned, just kind of floating around helplessly. And they'll pick them up, and Texas State Aquarium has recovery facilities. And for example, in 2021, 13,000 turtles were cold stunned, was the estimate, and only 32% survived that event. So how do we get better at targeting and finding these and kind of making the recovery and rescue more efficient? So they started using some drones to overflights because that's cheaper than manned aircraft, launched from the shore and from small boats. But trying to find a cold stunned turtle, as you see in this image, can be difficult, basically looking for a needle in a haystack through the viewfinder of the drone if you have connectivity and live video. So we want to automate that process and start looking at edge AI embedded on the drone to basically help. And then you could send out several drones and just get the information that you needed without staring at a video screen. So Oceans Unmanned does have some AI work with UAS back a few years ago when we were working with the ocean cleanup, mapping surface-borne marine debris. And we would fly in front of the recovery ships and kind of map the surface-borne marine debris. But that was done kind of after the flight. It wasn't done on the edge. And so we're taking that kind of concept of making hotspots of things you see on the surface and trying to make that embedded on the drone itself. And so we have a payload in development using a Sony ILX LR1, which is a drone specific. I think it's a 62 megapixel camera with a NVIDIA Jetson embedded in the payload, running a YOLO version 8 model to basically take a photo every few seconds, run that image through the NVIDIA on board, and basically answer a yes or no. And if it's a yes, then we will send that latitude and longitude of a positive sighting back through the ground control station to a moving map display on the ground control station, or on the vessel, or on the beach where the UAS operator is. And so you can get basically live heat maps of cold stunned turtles. And then we can direct the response vessels in a much more efficient manner to recover and rescue those turtles. So right now we're in the stage, we have all the hardware. Next step is to start training the model. The good thing about working in the marine environment is it is a continuous solid background. So if there is any anomalies, they're easier to detect. And of course, there'll be a lot of false positives originally, but as we train the model and get more and more imagery, that model will get smarter and smarter. So we're using two kind of different data sets to train the model. Images from NOAA fisheries. I think Melissa Cook is on this call. She's been providing from NOAA fisheries a lot of the real turtle imagery, but as you can imagine, it's hard to get enough images to train a model. So we've also been using a 3D printed model that was actually from an MRI scan of a real turtle that Texas State Aquarium ran through their MRI and then fed that information in a 3D printer. And that's the picture you see on the left there is actually a drone imagery of the model turtle floating. And in this case, we can go out whenever the weather is good and collect hundreds and hundreds of images at different sun angles, different water clarity, different altitudes, instead of just hoping you find the turtle that you need in regular turtle surveys that NOAA is supplying. So we can really supplement the amount of imageries we can collect to train the model. And I think that's probably my 5 minutes, so I'll stop for questions. And any turtle questions, Melissa can jump in and answer, since I'm not a turtle person. I'm an operationalist.
We have a question on the chat. I'm gonna direct you to answer all questions there because we need to continue. Thank you very much, Matt, for your talk. Now our third talk, is Alasdair Davies, who will talk about a breakthrough in low-cost sea turtle satellite tagging and telemetry.
Thank you very much. Okay, I am Alasdair Davies. I am a director of Arabada, a UK conservation technology organisation. And this is obviously a 5-minute snapshot of 10 years worth of work. So it's quite the milestone. There's a post on WildLabs about this, but it's a pleasure to kind of just show you today where we are with this very specific work. And we have been working to unlock access to satellite telemetry for sea turtles. And it started back in 2016. They were the development years when we had a real plan to look at how we could open source sea turtle telemetry hardware and make a difference in this space, which had very traditionally been an expensive arena to play in. So tags costing anything from $2,500 to $3,500 was very much the norm if you wanted to put a satellite tag onto the carapace or shell of a green sea turtle. And there was a post that I wrote back in 2016, 27th of May. I didn't even realize myself that it had been written 10 years ago until I Googled it the other day to put a few slides together about the breakthrough we just had. And back then, WildLabs was about 50 people. You could tell because you could type your username in when you created an account, and it would allow you to have your first name with no special characters. It was available. No one else had it. It was only about 50 people there. And you can see how far the community has evolved since then. And it's like this snapshot in time, even like where Arabada was an organization. It wasn't even formed then. It was just an idea that then got funded in 2018. But the text right there, the message in that article was, how could open source tech dramatically reduce the cost of tagging sea turtles? And then over the years, we started to work with partners around the world. This is the team in Principe in West Africa. And this is all of the kind of turtle beach team that do the nest checking every night. And we were teaching them how to use some of our very early prototype tags. And they were feeding back on their response, how easy it was, the performance at sea, etc., etc. And we started to build this partnership around the world. So although we started over in Principe, we soon started working with partners in Cyprus, with loggerheads, then to Cape Verde, where we did a lot of work with FMB, Guinea-Bissau, which is not actually even listed on here, but that was next too. Then over to Trinidad and Tobago with the leatherbacks. In the Maldives, we worked with rehabilitation teams. So that's with the Olive Ridley project. So turtles that were missing flippers, and you'll see a few of those later. To put tags on them, take them out of their rehab tanks and literally take them for a swim in the ocean. You're in a kayak and the turtle's there, and you return the turtle later to its home. And that allowed us to do this really in-house rapid testing to build a tag. This is what it looked like all those years ago. We were building enclosures out of ABS. We were putting in our little GPS board and just doing internestal clutches. So that's where you tag a nesting sea turtle, a female that will take the tag away for anything from like 13 to 16 days and then return to the exact same spot on the beach, which is brilliant because you can get the tag off, you can download all your data, and you can work with the turtles in a really fascinating way. There's not many animals that you can tag and they bring you that data back right where you're sitting exactly 13, 14 days later. But this was our kind of learning time to figure out, how do you get fast GPS at the ocean? How do you build a saltwater switch that works well and can transmit and deliver? And it all comes down to this. This is the anatomy of a telemetry tag. The tag in this photo is one of the leading tags in this space. It's a Wildlife Computers tag, which is a very excellent tag. And it's capable of a surface detection time of about 250 milliseconds and then a TX, so a transmission, in about a second. So when that turtle comes up to breathe, you get a very fast surface response and you get a message transmitted to the Argos constellation. And that message will give you telemetry data, i.e. where is my turtle? If you use Doppler to figure out where you are, a few satellites pick it up, or you send a GPS position back. And these tags have been the kind of go-to tags for a very long time. I still recommend everyone should use them because they are fantastic. But our job was to look at open sourcing that technology to make it more accessible for everybody in the turtle telemetry space. And so about a month ago now, we went to French Guiana with the team. So there's a lot of Arabardians on the call. There's Geoffrey Fournier, who is the lead engineer on this project, who is the brains behind what you see inside this tag. There's Grant, who did all of the enclosure work and came up with a process of how we pop these tags. And importantly, this tag runs with a 49 euro Arabarda SMD transmitter inside. And it's essentially everything we've learned over the years amalgamated into the tag that you see in front of you. So the antenna we used for saltwater switch materials, the battery chemistry, the supercapacitor inside, all that knowledge shared in an open source way to produce a tag that can, and this is the big screenshot I've been waiting for 10 years, deliver the same satellite quality Doppler data as a leading $3,000 tag. This is one screenshot of a tagged Olive Ridley turtle in French Guiana. We went there a month ago with WWF French Guiana to support their bycatch project. There are about 17 to 18 turtles tagged, and half of them were tagged with other leading manufacturers' tags and half with our tag. And a month later, we have this telemetry data tracking a turtle that nested, was tagged with the tag you just saw then. And the positions are fantastic. We're using the new Canis constellation, which gives us a good accuracy. And just this screenshot showing where the turtle has been over one month in space and time at sea with such a low cost tag is what we've been striving to do for those 10 years of development. It'll now go forwards and it will be used as a very low cost solution for turtles undergoing rehabilitation. The turtle is looking at you on the screen here is called Heidi. Heidi was the turtle that we worked with at the Olive Ridley project in the Maldives. And Heidi here would have a tag temporarily attached to a carapace, to a shell. You would put it in a wheelbarrow and you'd wheel it down to the ocean. You'd get in a kayak, she'd go for a swim with you and get some exercise. They'd do all of her kind of bloods and vet tests and she'd come back in again. And Heidi helped us test this tag back in the day. We now have it on active Olive Ridleys in French Guiana. And we are working now to make this accessible for you, the community. So the next stage is to make it accessible as a ready to deploy tag. So you'll be able to apply to access these tags. We're probably going to work with specific partners at first to make sure we can hold people's hand and make sure you have a good experience. But it's now going to become available, and it's very different to an idea 10 years ago. It's a physical, real solution, and the Arabada team are really delighted with the output and for being able to help the turtles in French Guiana, where they've had a 99% population decline, or the leatherbacks have, due to illegal drift nets and illegal fisheries and bycatch. So that is my 5-minute deep dive, or not deep dive, short dive into that news. I'm just really delighted to be able to share it with you all as a community, and really just showing where we've been after 10 years of WildLab community spirit to get here. So thank you very much.
Thank you very much. It's fantastic. Making technology more accessible for the community is really necessary. So thank you so much. I think we have a question. What is the cost estimate? I think you answered 45 euros, something like that.
Yeah, 45 euros for the transmitter inside. Okay. All of the components are open source. You can build this tag today if you went, looked at the parts that we have in our bill of materials. The trick is we want to take away that complexity and give it you literally in a box that you can order. You open it up, you have a magnetic switch, you turn it on, and it transmits, and it works. So the cost we are working on this very moment in time, because it's so fresh, you're looking at like data that the screenshot I took is from yesterday. So you're looking at fresh data from 1 month ago, and we've been watching that to ensure that everything's nominal, the tag's performing, everything's passing, which we have full confidence in now. So now our job is to look at what we can release it at in terms of cost to the community. But as you can see, it's way lower compared to other solutions, and that final cost will be the bill of materials that it takes to procure, to make it, a margin that we feel is acceptable to maintain it and sustain it. And then we will make that accessible, and we'll share the news via the WildLabs community as to what that tag costs and how you can get it.
That's great. Thank you so much, Al. We need to move to our ad break. So that's wrap up our speed talk sessions for today's Variety Hour. And during this Variety Hour meeting, we have an ad break where we share a few updates and opportunities from across our community before we jump into our long talk. So first, I'm gonna hand it over to Talia to talk about this.
Hi, everyone. Yes, we had a little preview of this already. I'll drop the link in the chat to both the 5 key findings highlight article from Vanessa and the full report. But basically, we have just released the 5-year trends report on the state of conservation technology, which originally came out in 2021. And this is thanks to responses from all of you on our annual surveys over the past 5 years. So it's just a really cool kind of snapshot of what all of you are telling us you're working on, what your needs are, what your challenges are. And we are excited to have it out in the world. So go have a look and please help us kind of get the word out. We're also going to be sending out our next survey to collect the 2026 data. We're switching to every other year now in the next month or two. So keep an eye out for that.
Thank you. Next announcement, Adrian, do you want to take it?
Yes, sorry for the camera. Hi, everyone. Nice to see you. Would like to share with you that we have finally opened the archive of ICTC 2026, which means that you can now access the video from the main stage speakers that are all available on our YouTube channels. But you can also find the presentation from each parallel rooms that will enable to go through the key topics that have been discussed. And of course, you can find, again, the photos of the event. Thank you.
Thank you, Adrian. And last announcement is that we have open applications for the 2026-2027 WILDLABS Group Leadership Program. Group leaders help facilitate discussions, share resources, and build connections around topics of interest within the conservation technology community. And this year, we are also exploring interest in launching 2 new groups, zoos and aquariums, on aquariums and human dimensions. So if either of those topics resonates with you, or if you are interested in helping lead an existing group, we'd love to hear from you. So I think someone has shared the link on the chat. And now we move on to our quiz, which this time is based on the World Ocean, because we celebrated the World Ocean Day on June 8th. So let's see how much you know about the ocean. So first question, which of these statements about the deep ocean is true? A, red animals are rare because red is highly visible underwater. B, most deep sea animals are transparent. C, red animals often appear black because red light does not penetrate to depth. And D, deep sea animals cannot perceive color. I'm seeing C, B, and some more B, C's. The answer is C. Red animals often appear black because red light does not penetrate to depth. Being red is one of the best camouflage strategies in the deep ocean. Next question. What is the largest migration on Earth? A, humpback whale migration. B, sooty shearwater migration. C, Atlantic bluefin tuna migration. D, daily vertical migration of marine organisms. I'm seeing a lot of these. Yes, you know it. Every day, billions of fish, crustaceans, and jellyfish and zooplankton travel hundreds of meters up and down through the water column. Next question. Approximately what percentage of the ocean floor has been mapped in high resolution? A, 15, B, 30, C, 50, D, 75%. Seeing A. Well, the answer is, according to my source, is 29%, about 29%. So B is the correct one, but it's pretty crazy that we still know less about the seafloor than we do about the surfaces of the moon and Mars. And that question, which marine creature produces one of the loudest sounds in the ocean relative to its size? A, snapping reams. B, krill. C, copepod. D, C, butterfly. I'm seeing a lot of A, and yes, you know that. And they produce this very extremely loud sound, which interferes with underwater communication and sometimes research. And last question, which of the following remarkable, no, not last one, which of the following remarkable abilities has been documented in the jellyfish? Navigating thousands of kilometers using Earth's magnetic field. B, surviving for years without food. C, generating enough electricity to stun prey. D, reversing its life cycle and becoming young again. C, D, C. What? The correct answer is D. The jellyfish, known as immortal jellyfish, can transform from its adult stage back into a juvenile stage. And this is the last one. During the deep sea expedition off the coast of Argentina, led by CONICET and Smith Ocean Institute, which famous cartoon character unexpectedly became an internet sensation? A, SpongeBob. B, Nemo. C, Patrick Star. D, Dory. I'm seeing some C's. A, B, C, A, C, D. Well, the correct answer is Patrick Star, due to this bat-shaped sea star, or Estrella de Mar Culona, that was found. So thank you all. That was our quiz for this variety hour. And now it's time for last talk from Kalindi Fonda. And So Kalindi, if you are ready, I will hand it over to you.
Hello, everyone. I'll talk to you about our project Spyfish Aotearoa, but also give you a little note on the International Conference, Conservation Technology Conference earlier this year. So let's jump right in. So what is Spyfish Aotearoa? It's basically an effort from Wildlife AI and the Department of Conservation in New Zealand to streamline and automate marine protected areas reporting with machine learning, citizen science, and experts. And so how do these surveys happen, or like how does reporting happen? Basically, this is how the surveys used to happen before, and it's the Department of Conservation in New Zealand records some videos with having bited, like drops down a frame with some bait, and they do this in more than the 40 marine protected areas around the country, and then there's multiple drops in each location inside and outside the marine reserves. Previously, this was done writing like on papers and on Excel sheets, and then things were uploaded on SD cards and hard disks. So it was, and each of these was happening independently in each of the marine reserves. And then the rangers would review these videos. Each video was 30 minutes per drop. So they were about 1,000 hours of videos per year. And the number that we want to get out of that is basically max number per species per video. So it's a lot of human time for one or a few numbers. And also, as mentioned before, it was all around the country. So if you want to find some number from a few years ago, you have to write an email to someone and so on. And then reporting would happen by combining all this info and sharing. This is our data. So as mentioned, there's like a bait in the middle, and then there's like fish that come to eat. So a few years ago, we had this idea that instead of there being a ranger doing the manual task of reviewing the videos, we could do a little bit of machine learning. However, it's not so easy, because what data do we put in this machine learning, and where does it come from, and how do we collect the new data, and where do we store it, and then how do you run this machine learning repeatedly, and how do you ensure that the annotation is like of good quality, and then also once you get the machine learning output, what do you do with it, and how do you put it all together? So our little machine learning project became this much bigger system, and I guess we're very excited because it's actually now working. Like it works from the beginning to the end, and we are in the iterative process of making it better and improving. So that's a great step of any project. So I think one thing that we learned from this is that already just cleaning the data and centralizing. So basically this upper part was a big win. So we did basically three things. We standardized, centralized, and normalized the data, which means standardizing, it's like naming standards, and then like in the field, inputting the data with apps instead of paper, and then like have video file names that are uniquely identifiable, and centralization, basically bringing all the data in one spot so that we can look at it and have dashboards. And then normalization, also making sure no repetitions. I'm sure you all know when you have Excel sheets and teams try to share these Excel sheets, there's all these like versions and things go out of hand very fast. And then we also, one benefit of that is that we can create like automated and manual error checking. One thing that we learned also is that it's good to have a drop ID, or basically our data being the video. We want to have a drop ID that is uniquely identifiable, because often when data is sent back and forth, what happens is people lose metadata and information about that video. But by creating this ID out of all the information, basically the metadata itself, that kind of helps keep track. And also if someone sends something somewhere, or someone finds a data, a file somewhere, we know how to trace it back. So also then when we have frames, we just have the same with like a frame second at the end. So one of my favorite things about cleaning up the data is that even before doing machine learning, you have wins. So for example, if you plot your inside the marine reserve and outside the marine reserve dots on a map, you can see what's happening. and I'm going to look at the chat, what people are saying. Do you see what's the problem here? There are actually two problems. So we have a little watch party here. So no suggestions. And also there's this hint here that this is bluish. No messages? Okay, I'll ask my guests here. So for example, these two are out of the way, like they're meant to be inside the marine reserves and outside the marine reserves. But then also this is the sea and this is outside of the sea. So basically like, yeah, observations on land, and it's all on land. So now we can see right away that something's off. And there's another question further down the line. So be ready to write your answers. Okay, so our pipeline basically has these steps like data collection, annotation, and counting process that is split into 3 steps: machine learning, citizen science, and experts, and then the data analysis and reporting step. And then I think community is really important, and this is something that came up a lot during the ICTC, and I think I've been thinking a lot since. Our process is basically the half-an-hour video goes into the machine learning, and then we funnel and give to each further steps less to review. So for the citizen science, we already select something, and then for the experts, even less. So the first step with the machine learning, we use a fine-tuned community fish detector model. And the community fish detector out of the box struggled with images from the top. But as soon as we gave it a few images, not that many, like a few hundred, it improved really fast. And now we are adding species on top in order to have both the fish detection, but also for some of the common species like snapper and blue cod, we can get the species right away. And then we get out of it like a selection of potential max ends on this half-an-hour video. And then that gets forwarded to the citizen science platform, where people watch a video and mention like the potential max end moment gets forwarded to the platform, and then citizen science review this and say where they see the max end and also which species it might be. And then we have an expert review step, which we do with BIGO, which allows for a very high level review of species. So for example, this can be like a half-an-hour video, and you just see all the snapper fast, and it allows for a much quicker both a review of the values so that the scientific number is correct, but also the feedback into the models. Okay, so now is the next task. This is a 10-second videos. Can you count the fish, max end fish on this video? I'll play it again. How many do you see? You can put it in the chat. So there's some 2s, there's a 3, some 3s, 2s, 3s. Oh, there's a 4. Okay, and then if we go forward, we can look at what our volunteers said. So some people said 5, some people said 3, then 2, then 3. Some people said spotty, some people said other animals. And so it looks kind of messy, but actually when we look at this, we see that the signal is that somewhere around second 1 or 0.1, there's a lot of activity. So we can then look at the machine learning results from there and forward it to expert review. When the video is simpler, it's less like there's less disparity. So this is one step where we're improving currently, because we don't think like this might not be the most efficient use of community, like of the community's time. So we're improving and learning from other projects on how to best see, like learn about where our model is making mistakes, and also to make it fun. What we've noticed is that the citizen science step is also an amazing tool to communicate with the community about what we do, and basically give people a chance to look underwater. And there's some rangers that actually ask to have some of their videos show always, so that when they talk to their community, they're like, yeah, look at there and see the videos that are there. So this is actually the max end frame. And if we look at the results, there's actually 4. So 1, 2, 3, 4. This is what the model did. So it got 3 right, and this one it got wrong, but it still found the signal. So we're doing like it's good enough to build on top of. The next step is basically to iterate on the model, make it better, and also create these scientific tools for rangers and giving back to the community, and hopefully create like where we've been building open source and modularly, but of course it's based on the example of the Department of Conservation, but we would love it to be accessible and useful for any kind of project, marine project especially. So for me, the important thing that I like to think about a lot is who do we build for and with, and for me, and our people are basically the rangers, the Department of Conservation, then marine experts, the volunteers, the local communities, and also the general public. And one thing that became clear to me over the last few months as our pipeline improved is that before a ranger used to have agency over the whole process. Like they would do the field work, they would do the annotations and then the reporting, and they knew from A to Z what happened and how. Whereas now, basically, they do the field work and then something comes out and they know that something is happening here, and this is a bit of a mystery. And it can be basically a thing where people might lose agency, but I feel like we can maybe transform this mystery box in like a fun box and use tools to create new ways to engage with the data that was not possible before. For example, the dashboard that we saw, but then also, for example, searching through the data and saying like, show me all the sharks, or as we saw, the citizen science as a local community engagement, and then easier collaboration with researchers and so on. I know some people have in their offices things like live, laugh, love. I don't know what, but this is what is my focus, which is to see people that we work with then enjoy the tools and find them useful. It's such a great feeling. And yeah, we do. I mean, for me, it's great when people tell me what's useful and how they use it. And so I try to communicate with people a lot. And there's a little data break. So this is an example of the model on our fish. And now on a little note on the International Conservation Technology Conference. So it was really, I think a lot of people found, I was talking to people, and some people actually used the words, oh, I feel like I arrived home. And it's because a lot of people we would meet and sessions we went to were really relevant to the work we're doing. And also just the networking and the exchange, everything was like useful. And the thing that really made a difference for me is that when I left, I had so much momentum to work on the project, but also because of the information and skills that we shared. And then also the interesting thing is there were so much future collaboration and sharing, like projects that we're working on now together, or even like you sign up to someone's newsletter and they say, oh, there's a conference there, and then you apply for that conference and you go there and meet again potentially. So it's really beautiful to see how coming together can make a difference. And then just a little a note on the workshop where people had to bend to look to assemble post-its. The idea was something like we build tech, but when we bring it back to when we take it to the field, something happens and it doesn't work necessarily. And so we had this opportunity to bring both technologists and NGOs and everyone in this spectrum, in this world, to kind of see what happens in these projects and what might go wrong or what goes well. And I think one of the takeaways that everyone has the same problems and we just need to talk to each other, which is pretty funny. But I guess a few themes was like just funding and capacity building, and then that we have to iterate with users and community, especially like planning, scoping, learning together, and knowing local people's, like understanding local people's expertise and seeing what they need. And then the reality of the field, sometimes there's no internet or the water gets into your camera. And also that having good people on the team can make things work out, like happen, but it's also like a great motivation. And of course, the need to learn from each other and share. One of my favorite titles for this was Scope Creep and Creeping Ants, because that's like the two different problems that happen, one in tech and one in the field. And yeah, this is the list of I like a bunch of post-it notes of yeah, I like them because they like the problems you don't get when you sit on the computer. And then I would just want to give 2 shout outs in the community. We organize some vibe coding parties, which is an opportunity to learn about the new AI tools that come. But if you are not interested in that, you can just come for a co-working session. There's like people from the conservation community. And then, of course, the community fish detector. They're looking for collaborators and data. So just check it out. And that's it. Thanks, everyone. And yeah, thanks a lot. And thanks a lot to WILDLABS.
Thank you, Kalindi, for this great presentation and for sharing all your enthusiasm. We are just on time. We'd like to say a huge thank you to all of our wonderful speakers today for sharing their expertise and inspiring work with us. I would also like to thank all of you for joining us, for your questions, and for contributing to the discussion throughout the session. You are all invited to stay for our after hours conversation. This is a more informal space where we can continue discussing today's talk, ask additional questions, and chat about anything related to conservation technology. Thank you again. We hope to see you again at next month's Variety Hour. Have a wonderful day.